Well, I actually was first introduced to mipsagargin or G202 probably about 4 years ago when we got involved with the drug in the phase 1 dose escalation study. And certainly I was interested in the compound, the way, the mechanism of action. And so we brought it into our center and ran the phase 1 trials in patients with multiple tumors, and was certainly impressed by the clinical activity, namely in patients with liver cancer. So G202 is an interesting drug. It has its active compound is a natural product called mipsagargin. For decades, we have known mipsagargin to have very strong cytotoxic effects both on cancer cells and normal cells. And what the guys at GenSpera were able to do with G202 is design a drug that has the potent cytotoxic compound of an analog of mipsagargin, and they linked it to a peptide that would not allow it to be active until it hits its target, which is PSMA. And taking into account the phase 1 study, which we saw some nice effects in patients with liver cancer, and knowing the fact that the target of G202, PSMA, is expressed highly in liver tumors, we chose to run this phase 2 study to see how patients with liver cancer would tolerate the drug and what the clinical activity of the drug would be. We enrolled a total of 25 patients into the study, so every patient got the drug. About a third of the patients in the study had some compromise in the liver function, and these patients are typically excluded from many clinical trials, but we allowed them to come into the study because we wanted to see what it is across the spectrum of patients with liver cancer. And what we found is that we saw a disease stability rate of around 65%. That means after two months of treatment, about almost 2/3 of the patients who enrolled in the study went on and got further drug. And typically what you would see is patients would progress in this meantime and succumb to their cancer. So a lot of patients had disease stabilization, and the time to tumor progression on the cancer was, for all patients on the study, was 4.2 months. And that's significantly better than patients who did not get any treatment, or when you compare it to other trials that have been done in patients who have failed the standard first-line treatment, which is Nexavar. So because it's a 25-patient phase 2 study with very encouraging results, with nice clinical activity, with time to tumor progression, the primary endpoint of the study of 4.2 months, considering that we took in about a third of patients with Charles-Pugh B disease, so their liver function could be a little bit compromised, I think there would be a strong rationale to go and evaluate this further in patients with liver cancer in a larger study, perhaps in a randomized fashion, to see what the comparator would be to no treatment and see if we see a true difference in terms of clinical activity.